All right, I'm going to jump right into this. We have a lot of information to cover between chapters 8 and 9, but what I did is for chapter 8 and 9, there's a lot of technical terminology. And initially, when I taught 1408, it was devastating for the students. Never mind comprehending the material as far as how a cell actually divides and the two different ways cells can divide, and how traits are passed on from parent to offspring. It was just the terms that were being used. Students were telling me, I don't even know what I'm reading. I've never seen this. So what I decided to do was put together kind of a glossary of terms that you're going to see in chapters 8 and 9. And so what I'm going to do is focus on going over these terms. These terms are not only to be applied to lectures covering chapter 8 and 9 that's in your text and in the notes, but also for your labs 7 and 8. Okay. So for lab 7 and 8, they're going to cover cell division, which is covered in chapter 8 in the lecture. You're going to need that information to help you out. So I went back and edited the PowerPoint, and I numbered the terms we're going to talk about, so you can compare it better, or better compare it with your notes or coordinate it with your notes. First term I give you is a life cycle. Okay, life cycle is simply defined as beginning at a point where we have two adults capable of reproduction. Okay. And then from there, it includes everything from the birth of an offspring to that offspring becoming a fertile adult themselves, capable of reproduction. So when you talk about a life cycle, we normally start right here. Okay, that's where it starts, and that's where it ends, and then the next cycle begins. Now, when you look at this life cycle, notice you see terms or see figures such as male reproductive cells or female reproductive cells, or these terms, 1N or 2N, or this term, embryo, okay, as well as terms such as meiosis. These are some of the terms we're going to cover as we go through this chapter, because you're going to see them throughout chapters 8 and 9, many of these in chapter 8. So here I have 2 and 3. In your notes, 2 and 3 have to do with reproduction. In general, living organisms are found to be capable of reproducing in one of two ways. Now, there are some organisms, and we will talk about this in chapter 9 when we talk about plants. There are some living organisms capable of both types of reproduction, okay? But these two types of reproduction are simply known as asexual and sexual reproduction, okay? There are two different ways of producing offspring of the next generation. Animals, such as mammals more specifically, okay, sexually reproduce. In other words, it requires a male and it requires a female. The male produces sex cells or gametes called sperm. The female produces sex cells or gametes called eggs. Okay, sperm fertilizes the egg. Eventually, that will develop into an offspring, the next generation. That's sexual reproduction. It requires two parents. One is male, one is female. The male has to be able to produce sperm. The female has to be able to produce eggs. You're going to hear these referred to as gametes as we move on. However, some organisms are capable of asexual reproduction. Now, asexual reproduction can actually happen two different ways. Okay, asexual reproduction could happen this way. What they're showing you is this guy right here. This is an amoeba. It's a eukaryotic living organism. This is a nucleus right here. This amoeba is asexually reproducing by simply going through a type of cell division involving mitosis, where one cell divides into two identical daughter cells that are amoebas. What's not shown here, because I forgot to edit the PowerPoint, is that there are multi-organisms that are capable of asexual reproduction. We will focus on plants in chapter 9. Okay, in chapter 9 we will talk about pea plants. Pea plants are flowering plants, and what a lot of people don't know is that a flower, it's not there for show or to decide whether or not it looks pretty or not. Flowers are actually reproductive structures. They contain two types of reproductive organs. One of the reproductive organs produces the egg. The other reproductive organ produces the sperm in that same one flower. Therefore, plants can also asexually fertilize. So asexual reproduction, there's always one parent. And sometimes that one parent will asexually reproduce simply by dividing into two identical cells. But as we'll talk about in chapter 9, sometimes that one living organism, that one parent, will produce an offspring by fertilizing itself with sperm fertilizing the egg. Okay, so these are the two types. Now, then you're going to hear a lot about this. When we go over the two types of cell divisions, what you're going to hear most for the first type of cell division, which involves mitosis, is you're going to hear a lot of talk of what are called diploid cells. Okay, the abbreviation for diploid is 2N. All right, but then once we move out of that, roughly about the middle of chapter 8, we're going to start talking about the production of gametes, which are sex cells, egg and sperm. Okay, we're going to focus on us mainly when we go over cell division. And when we go over the production of sex cells, gametes, called gametes technically, or egg and sperm, we're going to talk about how these gametes are haploid cells. And we use an N to designate that. Okay? Now, I'm going to start with haploid cells, the N. Okay? The N represents the number of chromosomes in a cell. The N represents the number of chromosomes in a cell. Well, if we're talking about a human cell, okay, when in the female ovaries, she produces gametes called eggs, or in the male testes, where he produces sperm, those cells are called haploid cells. We represent them with an N, or you can say 1N, okay, but that N represents the number of chromosomes. In this case, sperm and eggs are haploid cells, and we represent them with N, but if they're human sperm and eggs, the N represents 23 chromosomes. Okay, I forgot to put this down. Normally I write chromosomes like this. All right, Mr. Gaitan just joined us, and his little icon's in my way here. It's funny when y'all join the class, if y'all join after we started the lecture, it'll show me you've joined, and it'll put this little big, what do you want to call it, note that actually blocks my screen in part. I guess they want me to wait for y'all. So 23 chromosomes, okay? Got the S here. That's how I abbreviate chromosomes. However, the majority of the cells, okay, of a mammal, or specifically us, the human body, they are diploid cells, okay? When a sperm, which is a haploid cell, fertilizes an egg, which is a haploid cell, each cell is passing on 23 chromosomes from mom and from dad. So when that sperm fertilizes the egg, it results in the formation of a diploid cell. The 2, okay, the 2 represents the number of sets of chromosomes. I use that word of too much, I noticed. That's the simplest way I can water it down. So when you're looking at a diploid cell, the 2 represents the number of sets in a chromosome, okay? The N, again, represents the number of chromosomes. So whereas a human haploid cell, such as a sperm or an egg, okay, the N represents 23, then for a human diploid cell, okay, the 2N, what does that represent in regards to the total number of chromosomes or number of sets? Can anybody give me an answer to that? I need to figure out if y'all are awake, if I'm making any sense. So if the N represents 23 for a human haploid cell, such as a sperm or an egg, then for a human diploid cell, knowing that the number 2 represents the number of sets of chromosomes, right, 46. 46 total, okay? So again, I'm going to focus heavily on human cells. So I'm going to try to just say human for the most part through chapter 8. And then in chapter 9, though, I'll throw plants in, but hopefully you'll have a better grasp of things. So human diploid cells, which make up the majority of your body, and they're called somatic cells, okay? So the majority of the cells that make up your body, they're human diploid cells, and they're called somatic cells. And I'll start using somatic cells when we get into chapter 8, the actual topic. Okay, these cells have a total of 46 chromosomes. However, what we're going to end up doing is, as we transition from the first type of cell division involving meiosis to the second type of cell division involving meiosis, roughly about the middle of chapter 8, okay, you're not going to hear me say 46. Instead, what you're going to hear me say, and I'm going to put it up here, but you can put it underneath here. If you're taking notes, write it underneath where you put it down here. You're going to hear me say 23 pairs. Now I'm going to go over this, okay, during these terms that I'm reviewing. I'm going to go over this and I'll show you an actual chromosome map, which is called a karyotype. Okay, so the majority of the cells that make up the human body are diploid cells. They're 2N. They have 2 sets of chromosomes. The N represents 23, so there's a total of 46, but you're going to hear me say 23 pairs. Okay, and as I'll show you when I show you a chromosome map called a karyotype in a little bit, it has to do with a combination of the physical structures of the cell, including length, as well as the genetic information that they're carrying. Okay, we tend to mostly inherit genetic information in pairs, identical information from mom and dad. So these are diploid and haploid cells. When we go over the first type of cell division, when I actually get into chapter 8 and talk about cell division involving mitosis, we are going to focus on diploid cells, also known as somatic cells, make up the majority of your body. But when we get to the middle of chapter 8 and we start talking about a type of cell division involving meiosis, this is where we're going to talk about both diploid and haploid. Now listen carefully. The lab you've been assigned, okay, lab 7, these are the type of cells it's focusing on. You've been given lab 7. It goes over a type of cell division involving mitosis, okay? Okay, let me go back. So I have 4 and 5 and 6 and 7. Well, if you look here, 6 and 7 have to do with gametes and zygotes. Okay. So if you look here, it says gametes, but it says your gametes are what? Haploid, and what is it giving you? What I just went over. So this is another thing you have to realize. I've given you a glossary, but that doesn't mean these terms are separate from each other. Some of these terms actually go together. In other words, gametes are haploid cells. So sometimes they'll call them haploid gametes. In either case, whether we say haploid cell or gamete, what is in? 23. It's one and the same. Okay? In the human body, this is the only time you find haploid cells called gametes. Sperm and eggs, they're the only types associated with the human body. Okay? And I'll talk about this when I go over the second type of cell division in meiosis. How meiosis only occurs in the ovaries in females and the testes in males. That's the only site you see this type of cell division. It's a special type of cell division because I said the majority of the cells of our body are what in nature? Diploid. Okay? So this is more complicated type of cell division. However, okay, over here I have a zygote. Whereas gametes are denoted as being haploid cells. In other words, you can write an N. And I don't mind if you write down your notes, haploid gamete, one and the same if it helps, okay? Whereas gametes are haploid cells, so we can use an N to represent not only a haploid cell, but a gamete. Zygotes, okay, they're diploid cells. Now, in my day, you didn't hear about zygotes. What they would talk about is when a sperm fertilized an egg, okay, fertilization occurred, that fertilized egg would develop into an offspring, a multicellular offspring, specifically, eventually, a multicellular diploid offspring or adult, okay? But as scientists became more and more familiar with cellular processes, and especially cancer research, when it took off, they started getting more technical. There's a point in the life cycle where haploid cells are produced. Okay, these haploid cells are sex cells, technically known as gametes. Okay, so that's another thing I forgot to mention. The lay term for gametes, test, I'll use gametes, but just know they're sex cells, egg and sperm. Okay. And what happens is when the sperm fertilizes the egg, okay, we have a fertilized egg. But then what scientists say is that fertilized egg transforms into a zygote. The way I describe it to students is when the sperm fertilizes the egg, well, if you notice this little purple area with the N in each one, that represents the nucleus. When the sperm fertilizes the egg, what has to happen now is the DNA from the sperm needs to be combined with the DNA with the egg, resulting in the formation of one nucleus. Therefore, that fertilized egg transforms into a single-celled or single diploid cell known as a zygote. Again, a zygote is represented with 2N. 2 represents the number of chromosomes. N represents 20, or the number of sets of chromosomes, I'm sorry. 2 represents the number of sets of chromosomes. The N represents the number of chromosomes in each set. Therefore, we have 2 times 23 is 46. But we just put 2 in. Again, this purple area represents the nucleus. This is the rest of the cytoplasm with the membrane here. Now what will happen is this one diploid cell called a zygote will go through several rounds of a type of cell division that involves mitosis. This is what your lab 7 is focused on. It's focused on beginning here. That zygote will go through several rounds of cell division involving mitosis, resulting in a multicellular diploid organism that eventually becomes an adult. Well, more importantly, everybody, is the DNA contained here, the 46 chromosomes or the 23 pairs or 2 sets of 23. Whatever genetic information is contained in all of that DNA that forms all those chromosomes, that genetic information will be passed on to every cell of this multicellular offspring. This is another thing. Your labs are going to build on themselves. Okay? So from lab 7 to lab 8, we're going to get into lab 9s, 10, and 11. And I think it's 10 and 11 in particular. These are going to be DNA labs. I believe it's 10 that is a cancer gene lab and 11 that is a paternity-maternity lab. Both of them have to do with passing on genetic information. So eventually you have to understand the information we cover in chapter 8 and 9 to understand how it'll apply to the labs. So for you, your mom was diploid, your dad was diploid. But as I said, instead of saying 46 total chromosomes, you're going to hear me talk about them in pairs, 2 sets of 23. And we'll talk about as we go through chapter 8, how mom will pass on one of 2 sets of 23. Dad will pass on one of 2 sets of 23 in their haploid gametes. And then when the sperm fertilizes the egg, we reestablish a diploid organism. Okay? Now, so make sure, at the very least, when we finish going over this glossary, you know how these attach to each other, apply to each other. Okay, then 8. Well, going back to this one. What they're not showing you here is that a zygote goes through several rounds of cell division involving mitosis. I mess up a lot when I finish writing the word. It gets worse because I'm impatient. I'm like, hurry up already. Okay, so what happens, and they're not showing it here, okay, is this one diploid cell called a zygote will go through several rounds of cell division involving mitosis. Therefore, this one diploid cell will divide into 2 identical daughter diploid cells. Those 2 identical daughter diploid cells will in turn divide into a total of 4 identical daughter cells, which will in turn divide into a total of 8 identical daughter diploid cells. By the time we go from 1 to 2, and especially when we go from 2 to 4, we now have the formation of a multicellular embryo. So this is a multicellular embryo, a human embryo, and fertility doctors and scientists will actually number the embryo. They'll actually apply a number. So this is a human embryo. It's multicellular, and it's at what we call the 8-cell stage. It's made up of 8 diploid cells. Now, what are the two things that this eight diploid cells have in common, everybody? What are the two things these 8 diploid cells have in common? See 7 of them, one's hiding in the back. Can any tell me in relation to the previous slide, what I talked about? What are two common characteristics all 8, or features all 8 embryonic cells have in common for these embryo? First of all, they all came from where? What's the one cell they came from? They're all diploid cells, and what do we call that diploid cell that they all came from? All 8 cells came from one starter cell. What was that cell I said? It started with a Z? Right. So one common trait that all of these cells have in common, come on, they came from that one zygote. What we can't see now, but you're going to have to understand this because I did say it, the second trait they all have in common, since they came from the same zygote, has to do with the nucleus. Since all 8 of these cells came from that one zygote, that leads into the second trait they have in common, which is what? Has to do with the nucleus. Not the division, has to do with the nucleus. What's in the nucleus? Let me put it that way. They all came from that same one cell called a zygote cell, that diploid zygote cell. Okay. Now, what's in the nucleus of that zygote that they all came from? What's stored in the nucleus? Right. So then what do all 8 of these have in common then? If they all came from that zygote, then what must they all contain in their nuclei? 46 chromosomes, right? All made of DNA. Therefore, should all 8 cells have the same genetic information that was in this one zygote? Yes or no? Yeah. So I know it's broken up here, and I'll try to find a better figure. Again, the chromosomes passed on by mom and dad, the 23 in each gamete, the egg and the sperm. When the sperm fertilizes the egg, those 23 dad passed on are recombined with mom's 23 into one nucleus that is now diploid. Set of 23 here, set of 23, n plus n is 2n, for a total of 46. The way it works is, not only did all 8 embryo cells come from this one zygote, but the DNA, the total number of chromosomes, as well as the genetic information in this zygote, was passed on to all 8 of these. So, yes or no? All 8 diploid cells that form the embryo are genetically identical. Yes or no? Yes. They're clones. They're clones. This is important to know, okay? For some reason, the majors version of 1408, which is called 1406, the labs, they drive me nuts. Y'all actually have the better labs, okay? So some of the professors actually use the labs from this class for the 1406, but this is what I'm trying to get across. And listen, if you can't answer the question or you don't understand it, listen to the recording, because I'm just trying to, how do you say, get your minds running thinking about this information. And if you don't get it the first time, well, at least I'm pushing you a little bit so that when we actually start going over the 2 types of cell division and then we transition into how traits are passed on from parent to offspring, hopefully you'll think about, listen to this recording again, and you'll start saying, okay, now that we went over this in detail, this process, and I listened to the glossary, these terms, it's starting to make sense. So if you don't get it the first time, just keep plugging away at it and trying. Well, I keep saying chromosomes. And in textbooks, sometimes you'll see chromatin. Okay? And what do I want you to know we're talking about? Not just DNA, but what type of DNA? Eukaryotic DNA. Listen to me carefully. Prokaryotic DNA is circular DNA. So again, prokaryotic DNA is circular DNA. Eukaryotic DNA is linear DNA stored in the nucleus. Okay? Eukaryotic DNA is linear DNA stored in the nucleus. Okay. Why am I saying that? Because I'm going to compare these two together. But notice over here, this doesn't look circular. It looks like you have a bunch of licorice sticks or twizzle sticks or string. Okay. When a cell is not dividing, okay, when it's physically not trying to separate, okay, going from 1 to 2 cells, the DNA is described as being diffuse, spread out. Now, I didn't put this in your notes, but you can write this down under chromatin. Okay. The DNA is described as being diffuse, spread throughout, or another way you can say it is when a cell is not physically dividing into 2 cells, the DNA is loosely packed, taking up the entire nucleus. Whenever the DNA is diffuse, spread out, or loosely packed, we refer to it as chromatin. So you can think of it as chromatin as DNA that's very relaxed, diffuse, or spread out, or loosely packed. Okay? However, when a cell is going to physically divide, notice the difference inside here versus here, such as these little things here spreading out with these forming. When a cell is going to physically divide from 1 to 2, that diffuse or loosely packed DNA that we refer to as chromatin, it will tightly pack together. It will tighten up. And that's when we refer to it as chromosome. So now they're showing you how the DNA has tightened up. It's not occupying the entire nucleus. It's not diffuse. It's tightly packed together. And each of these strands we refer to as a chromosome. Now that being said, I'm going to throw in a third term in reference to DNA, but I'm going to throw it in when we actually go over cell division. Okay, I'm going to save it till then. Now, often what you hear in regards to DNA in general, okay, now let me recircle this again. Often here in reference to DNA in general is you hear the term chromosome, you don't hear chromatin much. It's because it's a lot easier for a scientist when they're teaching other people or students. It's a lot easier to refer to DNA when it has a shape or when it's tightly packed together in the form of a chromosome. It's just easier to visualize, easier to go over processes. In this class, it's going to get a little bit more technical, okay? But notice here, the DNA is linear. You only see that with eukaryotic organisms. So if you hear chromatin, you hear chromosomes, first thing you want to tell yourself is it's DNA, specifically eukaryotic DNA. Then add on the rest of that information. Okay. So here I'm going back one. We have 9 and 10. So like I said, I put the numbering in here, okay? But notice I have 11 through 14 on this one slide. I'm actually going to jump down to 14. Okay. 14, the term is karyotype. Now, online in the internet, I realized this morning I should have put karyotype as number 11 and then put the other 3. Okay. In person, it works better the way I have it because I explain what is meant by homologous, I explain what is meant by autosomes, which has to do with homologous, and then I explain what sex chromosomes are. So in person, it works better. I realized this morning, taking a closer look at it, maybe it would have been better to reverse it. But in either case, I'm going to start with 14, karyotype. Simplest way to define what a karyotype is, this is in lay terms, but you need to know karyotype going into the test, but this is in lay terms to help you out. Is a karyotype is a chromosome map. That fell apart. Wow, couldn't even spell that last word. That should be map. Okay, MAP, sorry everybody, chromosome map. Okay, now it's not just a chromosome map. It's a chromosome map most often it's a chromosome map of a 2N cell. What does 2N stand for again, everybody? Not a rhetorical question. What does 2N stand for? If I say 2N cell, what type of cell am I talking about? In general, not just a zygote, diploid. Yeah, diploid. So you're on the right track, Carter. You're on the right track. But in general, it's a diploid. All right. We don't want to say zygote right now because technically speaking, you're going to have a lab, a karyotype lab. I believe it's lab 9. And it will tell you in that lab the type of diploid cells that they will use to create this chromosome map. Okay, and they actually don't use zygotes, so that's why I said diploid cell, okay? So when I say a chromosome map of 2 end cells, just tell yourself in general, diploid cell. But yes, zygotes are. Okay, so what scientists and especially geneticists discovered is that when they can catch, say, a white blood cell in your blood, when they remove white blood cells from a blood sample taken from a person, they realized if they can catch that white blood cell, okay, in the middle of a type of cell division that involves mitosis, okay, that if they capture it at the right time, at the right stage, they can actually extract the DNA, say, from a white blood cell, extract it in the form of what we call chromosomes. Now, technically speaking, we cheat, okay? Because if you notice, it says each of these represents a pair of chromosomes. But then down here, it turns around and says, oh yeah, but also this right here, these guys right here, it tells you sister chromatids. I'll explain this later, okay? Remember what I said, often when a biologist or a scientist is going over DNA, when it tightly, DNA in general, they say chromosomes, even though it's not 100% true. The way they're applying it, it's just easier to visualize it and understand it, okay? So we do this with the karyotype right off the bat. I'll talk about what this term is during cell division. The karyotype is a chromosome map. And what scientists discovered, okay, is that when they take a blood sample from a person, isolate white blood cells, and then identify a white blood cell that's in the process of dividing from one cell into two, if they catch it at the right point when it's dividing, they can actually extract the DNA that's tightly packed together, forming chromosomes, and create a karyotype, a chromosome map. Now, I didn't look up the history of who first did this, okay? But what they did realize is that when they extract the DNA from a diploid cell at the right time of cell division, say from a white blood cell, they found out they can actually assemble a really nice map. But notice the numbering on the map. Top left corner, it says 1. Bottom right corner, it says 22. And then they show you an X and a Y. Well, what they realized is that when they catch a white blood cell at the right moment during cell division, not only are they able to extract this tightly packed DNA in the form of chromosomes, but they notice that these chromosomes have certain characteristics or traits in common to where they can pair them up. Okay? Also, what they noticed is part of the characteristics and traits that allows them to pair them up has to do simply with the length of the chromosomes. So what they started to do, okay, is ignore the fluorescent stain here. What they started to do is just based on length, someone decided that we are going to pair these chromosomes together and we're going to number these pairs. The chromosomes that are the longest, we're going to pair them together, put them in the top right corner, and we're going to call them chromosome pair 1. Okay? The next pair that are the second longest, we're going to call them chromosome pair 2, and so on down the line till we finally reach the point, okay, of being able to pair together the last 2 pairs, and we're going to number them 21 and 22. But if you notice, 21 and 22 here, they look like they're the same length. Well, there's a second characteristic they look at. I don't know if you can make it out here, but roughly about right here, it looks like you're looking at the upper shoulders or chest, and it looks like a little head on the top. Over here, it looks like you're taking a view from further back, a couple of arms and a torso and a head. I'm sorry if it sounds crazy. Well, what they realized is that chromosomes have what are called centromeres. On this pair, I'm trying to draw the arrow at where the centromeres are. Okay? We'll talk about this. Centromeres are the part of that tightly packed DNA or chromosome where you have a concentration of proteins. And we'll talk about what this centromere that's part of the chromosome that's made up primarily of protein, what the function is during cell division. But the second trait they look at is not only the length, they look for this little pinched in area which represents the centromere. You can see the pinched in areas here. You can definitely see the pinched in areas here. They're here. So you can see all these chromosomes have these little pinched in areas. These areas represent the centromere. So the second trait is not only the length, but are the centromeres roughly at the same point when you pair them up together? So that's the second trait that results in pairing them together. So if it just so happens that chromosome pair number 2, if one of these two looked a little bit alike one of these two, they would also look to see where is that little pinched in area representing the centromere. That would also help them further separate and delineate the difference between which ones form chromosome pair 1 versus 2. The third trait they focus on, right, they're mirrors of each other. I wouldn't, they're mirror images of each other. You could describe them that way, but you can also put them side by side. It's not so much, well, in other words, you can say they're mirror images of each other, but it's not like right hand and left hand. You can face them towards each other and they look the same. You can put them side by side and they look the same. This isn't true for all molecules. Some molecules, they're mirror images when you face them towards each other, but if you put them side by side, they don't look the same. Does that make a little bit of sense there, Zepeda? Yeah. Centromere we'll talk about, yeah, centromere is the second trait. We'll talk about the centromere more as we go, okay? I'll explain it. But it's a little tight waistband area here, okay? I don't know if you can see it on this pair here. You can see it here. You can definitely see it here where it's pinched in. You can see it roughly here. But there's a third trait. Scientists discovered they can use chemical stains. They used to use a stain called a chiasma or chiasma stain, which gave these chromosomes kind of a gray-black scale. But then they got fancier in the staining techniques, and now they use fluorescent dyes. So the third trait is the banding pattern after staining the chromosomes. Because these are very small. They look big here on the slide, but they're very tiny. I mean, you're talking at best submicroscopic. I'm trying to look at these things, okay? So what they did is, in order to avoid pairing together the wrong ones, okay, they also stained them. Because notice these two here, chromosome pair 1. If you actually look at the length, this one looks slightly longer than this one. But by staining the chromosomes, you can see the banding pattern, the pink, the yellow, and the green. The banding patterns between these are nearly identical. They're not perfect, but nearly identical. So that further ensures that they're pairing them together. Okay, so that's our karyotype. Now the way it works, okay, first of all, we have our diploid cells, which make up the majority of our body and are called somatic cells, okay? They have a total of 46 chromosomes, but when you describe the chromosomes in reference to a karyotype, we don't say 46, we say 23 pairs. And this is what I want you to get used to, 23 pairs. Human diploid cells, called somatic cells or zygotes, have 23 pairs of chromosomes, okay? Of those 23 pairs, at a minimum, I'm going to circle it here, and I'm going to circle it here. At a minimum, male versus female human, at a minimum, 22 will be homologous pairs. Okay? So on this chromosome map, chromosome pair 1, the largest, going left to right, top to bottom, down to chromosome pair 22, they are all homologous chromosomes. These homologous chromosomes we call autosomal chromosomes, or simply, whoa, where did that come from? Sorry, everybody. I swear I didn't do that, okay? These homologous chromosomes, I'm like, I didn't even move the cursor that way. These homologous chromosomes are called autosomes. At a minimum, when you compare a male karyotype versus a female karyotype, at a minimum, the first 22 pairs of chromosomes will be homologous. And these homologous chromosomes are called autosomes. They have a longer name, autosomal chromosomes, but often they go by autosomes. So let me try this again. So I'm going to put a line around these. But notice I'm not including the ones labeled X and Y. Okay? So when you look at a karyotype, which is a chromosome map, okay, that's when you should be able to identify not only that at least 22 pairs are homologous just based on size alone and where the centromere is located, but you should also know those homologous chromosomes are called autosomes. Now, what does homologous mean? If I ask you to define homologous, maybe I will on the test. How do you define homologous? What does it mean? Because you hear it a lot in the sciences, especially biology. Look at the first 22 pairs, right? I like to say identical in terms of structure, similar in terms of genetic information. So I define homologous as identical / similar structures or objects, okay? Listen carefully. What scientists discovered is that what you can't see in a karyotype is the genetic information. Not only are these homologous chromosomes homologous based on their length, where the centromere is located, and the staining pattern, but they're also homologous in regards to the genetic information they're carrying. As you scan, say, chromosome pair 1, wherever you find genetic information for a particular trait such as eye color, as you scan the other homologous chromosome 1 at the same location, you should come across the same genetic information for the same trait. Now, I'm not saying eye color traits are located on one. I'm just saying as an example. This is important. This is something else we will apply both in lab and lecture. Okay. So when you talk about a karyotype and you look at the karyotype and you come to understand what it's showing you, you realize at least 22 are homologous. And you realize those 22 pairs that are homologous, we call autosomes. That leaves one last pair. And I'm going to circle the 13. What scientists discovered is that sometimes it's tough to go by the length and where the centromere is located. But the staining pattern will still help out, and then genetic information definitely differentiates. But what they started to find out is when they created a karyotype, especially a male karyotype, they were always left with 2 non-homologous chromosomes, NON hyphen. They're not the same length, centromere is not in the same location, the staining pattern is not the same, and they are definitely not carrying the same genetic information. But what they did find out, okay, is that in particular, in reference to the Y chromosome, okay, especially this one, these last 2 chromosomes together, which I circled the whole thing, determine the sex of the offspring. Since these last 2 chromosomes collectively are said to determine the sex of the offspring, they're referred to as the sex chromosomes. I'm not going to write it out, but I'm pointing to number 13. They're referred to as the sex chromosomes. So when you look at a karyotype, and you're going to have to do this in lecture and lab, definitely lab, if I run into any problems getting the figure in lecture for the test. But when you look at a karyotype, okay, one thing you want to look is you want to see, are there 23 pairs of chromosomes to let you know, hey, appears normal, appears like it's from a normal human. Okay. Second thing you should notice is the first 22 pairs are homologous. Third thing, largest ones are in the top left corner. The 22nd pair is the smallest, or approximately the smallest in the bottom right. Then the next thing you should notice is the 23rd pair. The 23rd pair will let you know, was this a karyotype using cells from a male, or was this a karyotype using cells from a female? Okay. If you see 2 chromosomes that look nothing alike, and they're not labeled 23, but they're labeled, say, X and Y, then you know the sex of the offspring used to create the karyotype is male. If the karyotype were a female karyotype, what should this 23rd pair be labeled? Instead of XY, what should it be labeled? XX. I have to ask this question. I work with everybody, and if I don't find out during lecture and somebody doesn't come by during office hour, I don't pick it up in lab. I do pick it up by the test that I have some students that need to hear this, okay? So yeah, it would be XX. So technically for a female karyotype, there are 23 homologous pairs. It's just the 23rd pair is the sex chromosomes. Male karyotype, there's 22 homologous pairs. The last pair, which is the sex chromosomes, are not homologous. And we'll talk about these some later, okay? Cell division, simply put, cell division is where one cell, should be a 1, oh my God, let me try again. Let's not get fancy, right? Let me just put a line. One cell one cell divides into 2 cells. That's cell division. One cell divides into 2 cells. That's the simplest way to describe it. Now, is it always that simple? No. First of all, there's 2 types of cell division. The difference mainly focuses on what happens to the DNA in the nucleus and therefore the nucleus itself. So this figure says here, just in the past second, millions of your cells have divided into, especially in relation to the part of your skin called the epidermis. Okay, the epidermis is made up of cells called epithelial cells, and they're always going through a type of cell division called mitosis. A second area you could find cell division always occurring, okay, is in the ovaries of females and the testes of males. Now, for the testes of males, it can happen over a larger time span of the life cycle. For females, it's a more defined and shorter period of their life cycle, but you have cell division occurring. But the type of cell division is very different from the type of cell division that occurs with these epithelial cells of your skin or the epidermis. But here it says just in the past second. So here you're looking at the final stages of cell division. The cell membrane here, which was part of one cell, has pinched in. And it's going to pinch in all the way until they finally separate into 2 cells. So what you're looking at from the outside, okay, is towards the end of a process called cytokinesis. Division of the cytoplasm of one cell, okay, division of the cytoplasm of one cell by the cell membrane pinching inward, resulting in 2 cells, each with their own cell membrane, each with their own cytoplasm, and in that cytoplasm they'll have a nucleus, each with their own DNA. Okay. Listen, the type of cell division we're going to focus on, okay, is eukaryotic cell division. I'm not going to write that in because I can't write it. You write it in, okay? Let me ask you, in regards to where DNA is located, what's the difference in the location of DNA when comparing a prokaryotic cell to a eukaryotic cell? What's the difference in terms of where you will find the DNA, where it's located? If you're looking at a prokaryotic DNA, a prokaryotic cell versus a eukaryotic. Should be an easy question. Did y'all leave? Right, in a eukaryotic cell, you will find the DNA stored in the nucleus. In a prokaryotic cell, it's not stored in the membrane. It's concentrated in a region of the cytoplasm called the nucleoid. Because prokaryotic cells don't have membranous organelles. They don't have a nucleus. They don't have a rough ER, a smooth ER, mitochondria, or Golgi. They don't have any of those. They have ribosomes, okay, but they don't have that other stuff. They'll have flagellum, okay. Whereas with eukaryotic cells, they have membranous organelles. They have a nucleus, a mitochondria, rough ER, smooth ER, okay, along with non-membranous organelles such as the ribosome. So that's the difference. So when you hear me say mitosis and meiosis, I'm referring to nuclear division, and it's up to you to understand. I'm not talking about all living cells. I'm only talking about eukaryotic cells. They're the only ones that have a nucleus. Why am I saying that? Because I'm trying to explain what's going on here. First of all, cell division is also known as a cell cycle. That's what AKA stands for. In my generation, people knew what AKA stood for. I don't know how many years I had classes, both 1406 and 1408, and all my other classes had no idea what I was saying when I put AKA. They were just like, AKA, what the hell's AKA? Also known as. Cell division is also known as a cell cycle. Well, they're showing you here. This is cell division, but they're showing you why it's called a cell cycle. Okay? Cell division consists of a cycle of molecular events that begins with one cell and results in the production of two cells. Okay? Now, if you notice, this type of cell division they're showing you involves mitosis. Mitosis is not cell division, everybody. Mitosis is not cell division. What does it say mitosis is? Division of the what, everybody? Please answer that question. Right. Mitosis is division of the nucleus. Therefore, are we talking about prokaryotic cells at all? No, just eukaryotic cells. And what do you find in the nucleus of the eukaryotic cells? DNA, which is represented by these purple structures. Okay? Division of the cell, the entire cell, that's this term here. Notice it says cytokinesis is division of the cytoplasm. What is the cytoplasm? Somebody tell me, what is the cytoplasm of a eukaryotic cell? Since we're only focused on those right now. Tell me in the simplest way possible. And then it got very quiet. Come on, there are 11 of you here. Somebody's got to have an answer. What is the cytoplasm? Well, yeah, but where is it located? Where's that goo located, Carter? That's not a bad description, but where is that goo located? Yeah, interior or inside the cell. It's the intracellular environment, INTRA. It's made up of a solution called the cytosol, ICF, and organelles, membranous and non-membranous. So it's everything within the cell membrane or enclosed by the cell membrane. Okay, so therefore, division of the cytoplasm is where the cell, one cell, physically divides into two separate cells. But we want both those cells to have their own what, everybody? We want both those cells to have their own nucleus with the same DNA. Okay, so your lab is focused today. Your lab assigned on Tuesday is focused on this type of cell division. Again, why do we call it the cell cycle instead of cell division? How come they call it the cell cycle sometimes? Again, because it consists of a cycle of molecular events that begins with one cell and ends with two cells being produced. Okay. Now, cytokinesis is division of the cytoplasm. I'm going to jump ahead to this one. Cytokinesis is division of the cytoplasm. They're showing you an actual cell where the membrane is pitching inward. This one's easier. Animal cell, human cell. Okay, I'm not going to ask you for this stuff here. What I want you to know, cytokinesis is division of the cytoplasm. How does the cytoplasm divide it? Well, first of all, what they're showing you here is we already have two nuclei present. So mitosis is finished. We went from one nucleus in one cell to two nuclei that happen to be still in one cell. We'll talk about how at the end of mitosis, which is division of the nucleus, towards the end of mitosis, the cell membrane of that one cell will begin to pinch inward. It says cleavage furrow, but it's pinching inward. As the cell membrane pinches inward, it's going to divide that cytoplasm with the two nuclei of that one cell. It's going to divide the cytoplasm, okay, into two separate cells. But when the cytoplasm is being divided, that one cell ensures that each of the new nuclei produced at the end of mitosis are also separated into separate cells. Now, this type of cell division is a type of cell division that leads to that one diploid zygote cell dividing and forming every cell of your body. It's through this type of cell division involving mitosis. Okay? So we'll talk about it more. In your lab, I mentioned that the type of eukaryotic cell division that involves mitosis, initially this type of cell division begins with interphase, okay? And then at the end of interphase, we transition into the mitotic phase. We'll talk about this in lecture. I'm just saying this for your lab. This part I'm saying for your lab. The mitotic phase consists of two subphases: mitosis, division of the nucleus, 1 to 2 nuclei, and then it will end with division of the cytoplasm, one cell separating into two. Up here, the first phase, interphase, you hear a lot about interphase because interphase is divided up into three subphases. Again, this is for your lab. Divided up into three subphases. The most popular phase of interphase is S phase. Okay? This is where DNA synthesis, but more specifically DNA replication, you can also call it chromosome duplication, occurs. In other words, we want that one cell, okay, to make copies of all of its DNA to ensure that when it finally divides into two separate cells, each cell has the same DNA and therefore the same genetic information. This one here, okay, notice I have mitosis over here and meiosis over here. This one is very complicated, but both of these, both mitosis and meiosis, okay, focus on nuclear division. So please make a note of that. What they're showing you here is just the nucleus, not the cell. Okay? Both of these are forms of nuclear division. The majority of your cells that make up your body are produced through a type of cell division or cell cycle, including mitosis. Okay? That's where one nucleus divides into two identical nuclei. Notice the colors here and the links are the same. The blue and the red represent chromosomes made of DNA. However, in the ovaries of females, the testes of males, there's a type of cell division that I call the cell cycle plus meiosis. Meiosis, okay, is nuclear division, but what's happening is unlike mitosis where we begin with the diploid nuclei, resulting in two identical diploid nuclei. Notice the two ends here and up here. For meiosis, which occurs only in the ovaries of females and the testes of males, we begin with the diploid nuclei, but we end with 4 haploid nuclei. Now down here they say daughter cells, okay, but I'm not going to call them daughter cells. I avoid using that term for this type of cell division. You'll hear me say gametes or haploid cells only. I don't like to use daughter cells, and I wish they would stop doing that because it leads to a lot of confusion, not just for my non-majors, but for my majors. What we're going to talk about is that when mom and dad produce an offspring, they want the offspring to be diploid just like them. So we don't want mom passing on all 23 pairs of her chromosomes, and we don't want dad passing on all 23 pairs of his chromosomes. We only want mom and dad passing on one of two sets. In other words, only passing on 23 each, so that when the sperm fertilizes the egg, the offspring is a diploid organism, just like mom and dad. But in order to do that, we're going to talk about a second type of cell division, where when one cell divides into two, the nuclei goes from pairs of chromosomes to just a set. In other words, we go from a diploid nuclei to haploid nuclei. But then we're going to talk about how with this type of nuclear division has to go and undergo a second round of nuclear division. Okay, because there's something extra that needs to be done to ensure that the nuclei that are produced at the end of this type of cell division are only carrying 23 chromosomes total, nothing extra, and we'll describe it, okay? But mainly for now, understand when you hear mitosis and meiosis, those are types of nuclear division in eukaryotic cells. Mitosis here, that's going to be your lab 7, and then next week I'll assign you lab 8 meiosis. So put that effort and time studying for your labs, and there's some labeling too. And I say on the overview that with the figures I gave you, if I ask you to label phases in relation to mitosis or phases in relation to meiosis, For Lab 8, I tell you, you may not label every figure I give you. It's up to you to figure out which part of these figures refer to mitosis only, and where is it giving me extra that has nothing to do with it. Okay? So I'm really trying to bridge the gap to help you understand this topic. Are there any questions? Study these terms, become familiar with these terms. Please don't show up to class next week and you haven't studied these terms. It'll get really painful. Plus, in order for you to complete Lab 7, you need to study these terms anyway, because that will also help you with Lab 7 some, okay? All right, I'm going to stop the recording here.